So I'm obviously not here, so here's the little chat about enzyme function. What we're going to be attempting to figure out is how you can manipulate an enzyme in order to change its efficiency. So we'll talk about what efficiency turns out to be a measure of in a bit. It's actually going to be at the very end. We have to talk about what enzymes are first. To get there, helps if we remember, proteins are a product of gene expression. So I don't know if you can see the mouse, hopefully you can. You have a piece of DNA. Part of that DNA is going to contain a gene. So we find the data box. We'll open up the DNA there using RNA polymerase. What we'll end up creating is a piece of RNA. If we're expressing protein or a gene to make a protein, this RNA is going to be called messenger RNA. What I could then do is have a ribosome assemble itself onto the piece of mRNA at the appropriate start codon, and then it'll run through the A, P, and E sites until eventually we end up making protein. When we've done that, seen down here at the very bottom, hooray, job is done. When we build a protein, the part that's hardest is the primary structure. So we have to stick all these amino acids in order. There were 20 of them. Each one is unique because it has a different R group. Each of these is dictated by a codon, and a codon has 3 nucleotides. So if I had 100 amino acids, that means that this thing is at least 300 nucleotides in length. There's going to be more because the piece of mRNA is going to have sequences at the front and the end that are going to code for various things. Like we have a stop codon that doesn't show up, and we're going to have 5' cap and a 3' tail and things like that. What these amino acids will do with their peptide bonds is they will either form helices or they'll form sheets where the nitrogen and the carbon end up hydrogen bonding with each other, or you'll have areas that connect the two, so those won't form a helix or a sheet. Not all amino acids will form alpha helices or beta pleated sheets. These here will then fold 3-dimensionally to form what we call the tertiary structure. This is held together through ionic bonds, covalent bonds, hydrophobic interactions, and hydrogen bonds. All proteins have a primary, secondary, and tertiary structure. Not all of them have a quaternary structure, which is when multiple proteins from different genes interact with each other. The smallest quaternary structure you could have is 2. And obviously the more things you add on to it, the bigger the structure becomes. One of those examples of a protein is an enzyme. An enzyme is something that we call a biological catalyst. What a catalyst does, said in sloppy language, is it speeds up a chemical reaction. In reality, what it does is it takes one of these graphs, and if you look on the left side, it says reactants, right side it says products, and then you have this speed bump, this red speed bump. On the left side where it says uncatalyzed, it's of one height, and where it says catalyzed, it's smaller. What enzymes do is they increase the likelihood of a reaction occurring, meaning they make this energy barrier smaller. And if you make it more probable that a reaction will occur, what that turns out to mean is more reactions will occur. And if more reactions occur, we say it got faster. But in reality, it increases the likelihood of a chemical reaction occurring. This speed bump, activation energy, is manipulated by an enzyme. So why do we care? In order for all or any reaction to occur, this applies for everything and everything, even if it's not talking about enzymes, is we must have something called a decrease in free energy. And in class, I'll go through the math with you. Here, I'm not going to do it. But what we need to get is a reaction to occur that has a loss of energy. And if it doesn't happen, meaning if the energy is not lost, the reaction is impossible. If overall energy does not go away, the reaction does not occur. So here it gives you an example with this thing here, reaction 1, glucose plus an organic phosphate gives you glucose 6-phosphate. That reaction is impossible. So if I take glucose and add phosphate to it into a tube, mix it together, you will have a 0% chance of the reaction happening. It does not occur. However, if I were to take ATP, cellular energy, and stick it into a tube and see if it falls apart, the answer is it definitely will. So what I could do is if I were to combine this glucose plus the inorganic phosphate and react it at the exact same time as ATP falls apart. And if I could kind of manipulate the situation to ensure that the phosphate from ATP gets shoved onto the glucose, if I had an enzyme that allowed that to occur, what I would end up doing is increasing the likelihood. Would we expect this to happen? Sure, in reaction 2, the loss of energy, arrow going down means loss, is bigger than the upswing of energy for reaction number 1. So if I were to couple, meaning have reaction 2 and reaction 1 happen at the exact same time, if I add up the loss from energy from reaction 2 with the gain from reaction 1, I get reaction 3, which is overall a loss. This means the reaction will occur because it's required for all reactions. In this reaction here, it doesn't say the enzyme, but if you look, there's a little reaction arrow, and above it, you would actually write down the name of the enzyme, which is glucokinase or hexose kinase, depending on who you talk to, because it has a few different names. But the enzyme allows this thing to occur. Glucose and ATP are what we call the substrates. The glucose 6-phosphate and ADP are the products. The enzyme itself is not part of the reaction, so it is what it is. The thing is, with these enzymes, because they're proteins are picky because their shapes are picky, is they are very specific. Meaning, this enzyme here, L-alanine oxidase, will only work for L-alanine. L stands for left-hand version of it. It deals with something called stereochemistry. So this shape, this weird puzzle piece shape here, is what we call the active site, meaning it's the part of the enzyme that does the chemistry. It's the part that allows these reactions to occur. So, because the protein's shape is specific, that means the shape of the active site is going to be specific, which means if the substrate does not fit the active site, the enzyme is not going to do its job. So if I were to use something called D-alanine, or the right-hand version of alanine, it doesn't fit the active site. What does that mean? Nothing will occur. They are both alanine. Chemically, they are built the same way. The difference is they are spun differently. They have a different shape to them. And the result is nothing occurs. Enzymes are incredibly specific. The thing is, I can manipulate them rather easily, because if I can manipulate an enzyme, if I can manipulate the shape of a protein, I can manipulate the enzyme. So the way that this turns out to work is, normally what you'd have is your substrate, and it binds to the active site. In reality, the substrate actually kind of induces the shape of the active site, meaning here they look like they fit. In reality, they wouldn't do that. The active site would be a slightly different shape, but when the substrate comes in, it manipulates the environment around the active site, which will cause the active site to change its shape so that the substrate fits. And it's called an induced fit model of enzyme function. But if you look, here it doesn't work. This is doing a lock and key. The substrate fits the active site, which means this substrate is meant for this active site. But what would happen if I manipulated it? Meaning if I had something that could fit into that active site, well, then it would compete, meaning the substrate and this other thing, this inhibitor, this stopper of the enzyme, they're going to fight for this little slot. And the result is the enzyme's not going to work as great. How do we know? Because we have something inhibiting it, or a competitive inhibitor. Competitive meaning the substrate and this other thing, this inhibitor, are fighting for the same thing. They're both fighting for the active site. But that's only one way I can mess with this. I could also just mess with the protein other ways. So there are things that we call non-competitive inhibitors or allosteric inhibitors. That would be something else that can bind to the protein, and by binding the protein, I'm changing the environment. I manipulate all the bonds, so the covalent bonds, the hydrogen bonds, the ionic bonds, the nonpolar interactions, and that will warp the shape of the active site. And it can warp it enough that the substrate doesn't bind. And the result is two ways. I can sabotage by blocking the substrate, or I could sabotage by altering where the substrate is supposed to go. So it's like behind the scenes manipulation. But for the sake of also saying it, what we could also have is, with this non-competitive inhibitor, there are some things called prosthetic groups or coenzymes that are needed to make this enzyme work. Meaning, if I didn't add this other thing to this allosteric site, allosteric, so other location site, the enzyme doesn't do its job. And that's usually the role of something that we would call a coenzyme. They're needed to make the enzyme do its job. So this is the only way we can mess with enzymes. We just have something in the way of the active site, or we can mess with it on an allosteric site? Well, of course not. We still have the other things, because after all, they're still proteins. So if I change its pH, I mess with ionic bonds and hydrogen bonds, which will denature the protein. If I mess with the temperature, I'll destroy the hydrogen bonds and the ionic bonds, which will destroy the protein or denature it. If I add salt to it, I'll change the nature of the bonds, which will denature it. If I add it into a different solution, which now will make all the interactions different, because if I take a protein and throw it into oil, well, oil is nonpolar, so suddenly those nonpolar amino acids can come out of the woodwork and interact with the oil, and that will change the shape of the protein. So I have lots of things I can do. I can add inhibitors in some way, shape, or form. I could add coenzymes. I could also change the pH or the temperature, or add salt, or change what I'm dissolving it in. Why all this matters is we could talk about enzyme efficiency. Through a whole bunch of math that I don't want to do, and you don't want to do right now either, what we can do is we could figure out something called Michaelis-Menten, or use something called Michaelis-Menten kinetics. Kinetics is how quickly reactions occur. So if you look here, this black line is telling us for any given enzyme, if I change the substrate concentration, how quickly, VI, how quickly is this reaction occurring? And namely, it's at the start, because eventually we run out of substrate. So if I start with low concentrations, the reaction goes so fast. But if I increase the substrate concentration, the reaction goes faster. Makes sense. At some point, it maxes out, or what we call Vmax. The reason why it maxes out is you only have so many substrate, or so many active sites, and substrates can only get in and out so fast, and the active site can be cleared out only so fast. So we have a limit as to how quick enzymes can do their job. The way that we judge the efficiency of an enzyme is how quickly do you get to Vmax. The way we do that is I take my Vmax height and I cut it in half. When I cut it in half, so half of Vmax, I go to my graph and I look down. So what is the substrate concentration to get halfway to my maximum point? That halfway point is called Km, or the Michaelis constant, or the Michaelis-Menten constant. The Km value, the smaller this number is, the more efficient an enzyme is. So the quicker we can go from 0 to 100, the faster it is, or the better it is. How do we judge cars? We talk about how quickly it goes from 0 to 60 miles an hour. Well, why can't we do the same thing with an enzyme? How quickly does it take you to go from 0 to maxed out? That is your lab. It's, you need to create a plot that looks like this to then figure out Km. How do you do that? I need to figure out the concentration, or use one concentration, figure out the velocity. Use another 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 concentration, figure out the velocity. Another concentration, figure out the velocity. Another concentration, figure out the velocity. So on. This graph is a product of 5, 6, 10, 20, 50 different experiments. It's not one experiment of one enzyme concentration at one substrate. It's one enzyme with a whole bunch of different substrate concentrations to produce this graph. That's your task.